Okay guys, so last time we went over all of our passive transport mechanisms. We went over simple diffusion, we went over facilitated diffusion, and lastly we went over osmosis. So now we're going to start on the active transport part of it. So it's a little bit different, active transport compared to passive. Active transport is going to use energy. So all the other ones we went over, no ATP was required. This one, we need ATP. And we're going to move these molecules against their concentration gradient. So if you remember all the other ones, we were moving from high to low concentration. For this one, we're moving the opposite way. We're moving low to high concentration. And since we're moving against, that's why we need the energy. That's why we need the ATP. So our most common form of active transport is going to be the sodium-potassium pump inside of our cells. So what happens here is we are going to move sodium, and this is a carrier protein right here. We're going to move sodium out of the cell. Okay. Why do we want to do this? Well, sodium, like salt, anywhere there is salt and sodium in your bodies, that's where water is going to go. So wherever salt goes, water follows. Well, that's bad for our cells because if we get a lot of sodium inside our cells, yeah, our cells are going to blow up like a blimp. Okay, they're going to get real big and explode. And we don't want that. So here's what happens. In our carrier protein here, we are going to fit 3 NAs into our protein, and we are going to transport them out of the cell. Now, Na is in low concentration already inside the cell, but we're transporting it out. So we're going from an area of low concentration to an area of high concentration. That's why this is called active transport. In doing so, we need to use ATP over here, our energy guy, to get that Na out of the cell. Oops. And then once our Na leaves, we are then going to bring potassium into our cell. Okay? And why we're bringing potassium into our cell is to try to make up for those lost charges we just did by bringing our sodium out. Okay? So we brought the sodium out. It has a positive charge. We're going to bring sodium in, which also has a positive charge, try to even it out a little bit. Now the sodiums have a positive charge, like we said, and we're going to transport 2 of them in. So 3 NAs go out, 2 sodiums go in. Okay. Okay, so you guys can see them going in there. Now here's your difference in pictures. You guys can see passive transport, we're always going from high to low. Active transport, we're going from low to high. It's the opposite way. Now with active transport, we are going to get some electrical charge distributions in our cell because we're going to mostly be moving ions into and out of the cell, and these ions have some kind of charge. So normally our cells have a resting membrane potential, and that is the voltage across our membrane. So on one side of our membrane, it's going to be one charge. On the other side of the membrane, it's going to be another charge. Okay. So with these 2 forces, we get an electrochemical gradient. Okay, that creates our resting membrane potential. Our potassium channels. So along with having the sodium-potassium pump, that sodium-potassium pump, it transports sodium and potassium, and that pump can transport both of them. It takes sodium out, brings potassium in. Okay. Now, since it's bringing potassium in, and this is a pump, this tells us on the outside of our cell we have a low concentration of potassium, and inside of our cell we have a high concentration of potassium. While this is happening, maybe somewhere else in the cell, somewhere else in our cell membrane, we have regular potassium channels. Okay? And the potassium is in high concentration in the cell. Well, these other channels for potassium, they're leaky. Okay? And that's a weird word, but they're leaky. So what happens is some potassium will just diffuse, still through a protein channel, out of the cell, and that does not require energy. And don't forget the potassium has a positive charge. And since the potassium has a positive charge and we're taking that potassium out of the cell because it's leaking, that leaves our cell to be slightly negative of a charge, okay? So that means the inside of our cell is a negative charge. The outside of our cell is slightly positive, okay? The sodium pumps, okay, the sodium-potassium pump, we got the sodium going out, but there's also sodium channels in the cell, okay? But they are not as leaky as the potassium ones. So in our cells, we have a resting membrane potential. The inside of our cell is going to be slightly negative, and the outside of our cell is going to be slightly positive. Okay. We're going to talk about proton pumps. Okay. Now, a lot of our organisms have proton pumps, and it says there are plants, fungi, bacteria. We have them. Okay. So, moral of this is that all three of our domains have these proton pumps. Bacteria has them, archaea has them, and the eukarya, which is us. So given that, since all three domains have proton pumps, this tells us that go back far enough, all three of our domains, we probably had a common ancestor since we have the same exact proton pumps in our bodies. So here's a little picture of a proton pump, and I will expand this a little bit more later on. But we have a proton pump. We have inside the cell on the left side, outside the cell on the right side. And what we're doing is we are taking this proton, a positively charged hydrogen, that's what a proton is, and we are pumping it out of the cell. So we're going from low inside the cell to high outside of the cell. This requires energy. So why do we need proton pumps? Well, proton pumps are going to help us with co-transport. And what this is going to do is it's going to help us to bring something else into the cell. So what we did with this proton pump is we pumped the protons outside of the cell. Okay? So what we're going to do is after we have that proton pump outside, or that proton outside of the cell, okay, that proton, which we just required ATP to use, come back on, lights. They do not want to come back on. Sorry, guys. There we go. Okay, so we pumped the protons to outside. We went from low to high concentration. And now what we're going to do, and I wish this picture had a little bit more sucrose available, but we are going to bind the hydrogen into this protein over here. In binding that hydrogen, it helps us to transport sucrose across our cell membrane and take sucrose into our cell to be broken down into glucose and then used by the mitochondria to supply ATP. Okay? But if you notice in this sucrose H plus cotransporter protein, there is no ATP needed. So this tells us that this is a type of facilitated diffusion. The sucrose is in high concentration outside of the cell, and it's moving to an area of low concentration inside of the cell. Okay, so this is both active transport and facilitated diffusion. The proton pump is active transport, and then the cotransporter protein is going to be our facilitated diffusion. Okay, so that is the end of active transport. We're going to go over things being brought into the cell and leaving the cell. So this is just an example of active transport. We went over the mechanism of active transport. Now we're going to go over how it applies to our cells. So we have exocytosis. If you guys see the prefix exo, that is exiting or leaving the cell. So what's going to happen here is we are going to have a vesicle, like the ones we used for making a protein with the rough ER and the Golgi apparatus. we're going to take a vesicle. It's going to be filled with something, whatever it is. And that vesicle is going to fuse with our cell membrane, and then it's going to release our contents out of the cell membrane. We are also going to have, along with bringing things out of the cell, bring things into the cell. So exocytosis is things exiting or leaving the cell. Endocytosis is going to be things entering the cell. And there's three different types here. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is cell eating. This is when the cell is bringing in food. Pinocytosis, this is when the cell is bringing in some type of solvent, usually water. Skip this guy. Okay. So, I like the picture on the right better, obviously, the nice animation, rather than the one taken with the transmission electron microscope here on the left. But you guys can see that we are forming, right here, we are forming a vesicle. That vesicle is bringing the food into it. And then once that vesicle is formed, it's actually going to pinch off. So the phospholipids are going to pinch off, and then right here, right here, oh, that is a horrible color to use for this. This is black. Right here and right here, once this vesicle closes in on the food, these two parts will come together, and our phospholipids will once again close our cell membrane so that we don't let a whole lot of whatever in. So again, that is endocytosis. Here's pinocytosis. There's some solvent particles coming in, and once again, they're being put into a vesicle. Okay, the last one is receptor-mediated endocytosis. This is when we are going to have some type of receptors on our cell membrane, probably a carbohydrate of some sort. And these are going to be brought in and allow whatever to come in with them. Without the receptors, we're not going to allow that vesicle to be formed, okay? And you guys can see these little purple things here are being put into the receptors. That is what is catalyzing this whole reaction, this whole thing. Okay. All right, so now we're going to go over cell-to-cell communication, which is so much fun. Not really, but we'll get through it. Okay, so we're going to talk about how cells communicate to other cells and how they also communicate with kind of themselves. So gap junctions and plasmodesmata, we talked about these last chapter. These are ways that our cells, through gap junctions, are able to communicate with other cells. And plasmodesmata, this is ways that plant cells are able to communicate with other plant cells. Okay, it's all the same organism. It's still the same plant. It's just from cell to cell. Oops. Okay. For this one, guys, this is just talking about signaling. Paracrine signaling at the end there, stimulated by the pancreas. So we're going to talk about how all these signaling things happen and how we get these messengers in order to help us with cell signaling. So there's three different ones. There's short-term signaling, and there's two different types of that there. There's paracrine and synaptic. And then the third one is going to be long-distance signaling, when we have to maybe have an endocrine cell and go somewhere completely different in the body, very far away. So these two are local, and then the third one is long distance. So here is a local one. You guys can see we have cells, okay? They are surrounding this original cell, and maybe that cell has some kind of hormone in it or some kind of signal. And these secretory vesicles, they're taking those hormones and giving them to other cells. What are those other cells going to do? Well, it's going to give some type of response for those hormones. This is how our nervous system works. Okay, it's just one cell going to another cell, and they keep delivering the message from cell to cell to cell. Here is synaptic signaling. Okay, so this is how our neurons work. They go from one side of the cell to the other side. Okay, so this is like one side of the cell of one cell, and then this is the other side of a cell of a completely different cell. But you guys can see there's neurotransmitters, hormones, whatever they are. They're going to that target cell, and then once they're there, that cell will give some response all the way down this whole synapse here, and it'll go down the axon of this cell into the dendrites, and then it'll go to another cell and do another cell, okay? So we already talked about the hormones. For the long distance ones, we're going to use these hormones, and they're going to be put into the blood, and then the blood is going to take them to wherever they need to be in your body. Okay? So you guys can see right here, they're traveling throughout the circulatory system. Okay, so cell signaling. That was the easy part beforehand, talking about the hormones. Now we're going to get into the molecular part of it, which is made up of three parts: reception, transduction, and then a response. So here is the first step, which is reception. We are going to have a receptor, some type of protein that's put there in purple. And then we are going to have a signaling molecule, and this is going to start the whole cascade of everything. So that signaling molecule, most of the time we're going to call it a ligand, L-I-G-A-N-D. Okay? And what's going to happen is once we have that protein activated by the signaling molecule, by the ligand, it's going to relay these molecules, and that is called transduction. It's going to go from one molecule to the next to the next. So this is our cascade of different molecules being used in order to deliver some type of message. And once that message gets to where it needs to be in our cell, then we are going to have our response, our activation. Okay? Okay, so we already talked about the ligand. The ligand and the receptor, they are going to bond together. It almost fits like an enzyme and a substrate. The ligand is very specific. We're only going to have one ligand that matches any receptor. After that happens, we're going to have a change of shape in that protein, in that receptor, and then that's going to start all of our signals going from one thing to the next. So two main types of membrane receptors. We're going to have a G protein coupled receptor and a ligand gated. So just two different ways we can get this done. So a G protein, we're going to use GTP. It's another molecule we can use for energy. GTP, it's going to have 3 phosphates, just like ATP does. And then we are going to transfer it into GDP, which is going to have 2 phosphates. That D stands for diphosphate. And when we break off that phosphate, we get energy out of it. Just like when we convert ATP to ADP, we also get energy out of it. So here's a picture of the G-coupled protein receptor. So once again, we're still going to have our signaling molecule coming in, bonding to our protein. We need to bring GTP in. Okay? Once we bring the GTP into this other protein, whatever message that GPCR protein is trying to convey, it's going to deliver that message to our G protein here, and then that protein is going to take it somewhere else. Okay, in this example, it's taking it to an enzyme. And when it binds to that enzyme, you can see the activated enzyme. It's going to change its shape, so it's going to have a nice active site at the bottom there, and that will give some type of cellular response. Without GTP, without that energy, nothing is going to happen. The other one is going to be a ligand-gated ion channel. Same overall goal. The overall goal is to get some type of response, okay? So what this one's going to do is we are going to use ions, and they're going to be able to pass through, but in order to pass through, we have to use a ligand, some type of signaling molecule. And in this one, it's always going to be a ligand. Okay? So what happens is the ligand is going to bond to that protein. When it bonds to that protein, here in step 1, that protein is going to change its shape. So ligand bonds to the gated protein. That protein's response to that ligand is to change its shape. And then, once it changes its shape, we can let some ions through, okay? But there's going to be a point where, hey, we have enough ions. We're good, okay? At that point, the ligand is going to disconnect from that protein, and then the gate will once again close. The protein will once again change its shape. So that's ligand gated, okay? A little bit different there. Okay. We're going to go over receptor proteins now. These are going to be things that are going to be in our cells. Okay. There's a bunch of different ones. We'll go over a few of them. But testosterone, this is going to be one of them. It's a hormone. We can use a lot of different hormones in our body as receptors, okay? And chemical messengers, they can help us out. So here is an example. We're going to bring a hormone into the cell. It's going to, and it's testosterone all the way at the top. We are going to bind that testosterone to a receptor, lights go out, protein. Okay? Hello. There we go. We're going to bind that hormone, that testosterone, to a receptor protein. When that receptor protein has the hormone bound to it, it will then have some kind of response, which is, in this example, going to our DNA. Okay. When it goes to our DNA, it then triggers DNA to do something. So remember last chapter, guys, we talked about DNA making proteins. This is now we're getting into gene expression, how DNA knows to make them proteins. DNA isn't just like chilling in the nucleus and it's like, hey, DNA, what you going to do today? Give me 10 minutes. I'm going to start making proteins then. It doesn't work like that. It doesn't just decide like, hey, I'm going to make some proteins right now. Just give me a minute. I'm going to chill here for a minute. I'm trying to play some video games, and I'm going to get to them proteins soon. It doesn't work like that. Right when the testosterone comes in, that's what triggers the DNA or another hormone. It doesn't always have to be testosterone to make a protein, okay? Without that, without this hormone, without this catalyst to bind to the receptor protein and get things going, DNA is not going to give the instructions to mRNA to make a protein. Okay? All right. So we're going to go over the cascades. There's different types of ways we can go to get this response by our cells. Okay? Your book describes it as like falling dominoes. All right? What happens here, guys, is we activate a lot of different things in the cell to get some type of response. So the further into the cell it is, the more things we have to activate. Okay, it just makes sense. These things aren't going to be like doing a whole lot of moving. So, you know, the further it is into the cell, the more stuff we need to do. Okay, so we have phosphorylation and dephosphorylation. Phosphorylation, very simply, adding a phosphate to something. Dephosphorylation, yeah, you guessed it, it's the opposite. It is taking a phosphate away from something. So what we're going to do here is we're going to use these things called protein kinases. And what they're going to do is they're going to be dealing with ATP to transfer our phosphates. So the protein kinases are originally going to be dealing with ADP, and they're going to phosphorylate our ADP. And in phosphorylating it, the ADP is going to get turned into ATP. So that's our goal of our protein kinases. Now, if we look at this diagram here, we have our ligand, our signaling molecule, being bonded to our protein receptor here. And all these different kinases, we have kinase 1, kinase 2, okay? What they're going to do is in transferring our ADP to our ATP, it's going to give these protein kinases energy, okay? So you can see the ATP gets transferred into ADP. Where did that phosphate go? Well, it went to the kinase, okay? So now the kinase has it, all right? And it just keeps going in and in further. So we have different proteins, and after it gets so far into the cell,
Teachers, may I have your attention? If anyone tried to call the Kino residence, please call Tina at 5301. Thank you.
After all that happens, we have our inactive protein. Our last kinase is going to activate that protein, and then we will get some type of cellular response. Okay. So the opposite of a kinase is a phosphatase. They are going to remove the phosphates from our protein. So we need the kinases to add the phosphates, and we are going to need the phosphatases in order to take the phosphate away. Okay. So both of these are able to be reused many times. Okay. They're both enzymes. Okay. Anything that ends in ASE is an enzyme. Right. Along with these, we're going to have secondary messengers. The ligand, that's our first messenger. Okay, he's outside the cell. He binds that receptor, and we start the cascade that will hopefully give us a response in the end. Okay. Secondary messengers, these are small, non-protein. They are polar. Okay, so they're able to combine with water, and they're going to be all throughout our cell. Two main ones we're going to use is cyclic AMP, abbreviated CAMP, and calcium ions. So these are our main ones. What they're going to do is they are going to activate our kinases. And why that's good is, well, we can start our cascade. We can start our phosphorylation through that whole process. And then, at the end, hopefully get a response. And that's our goal. In this, the middle one, guys, adenylene cyclase. Sorry, voice is starting to stutter a little bit. So this is going to be an enzyme. What it's going to do is it's going to convert ATP to CAMP. Now the AMP, almost the same as the ATP, but the difference between ATP and AMP, the TP has 3 phosphates. The MP means monophosphate, which is 1 phosphate. So in conversion of ATP to CAMP, it actually loses 2 phosphates from it. And then here is our whole cascade. Starting our whole cascade. We'll have a little bit of a bigger picture later on. But we have our G-coupled protein here. Okay, so this was the first one we went over before we went over ligand gated. But we get our GTP in there. And now we have the adenylene cyclase. Once this bonds to it, the adenylene cyclase will change its configuration. It will create an active site there. Okay. And in creating that active site, our next response, well, we're going to have this secondary messenger, the CAMP, and then it's going to start the kinases through that phosphorylation cascade. And again, our goal is to get, at the end, the cellular response. So we're just adding more characters into this. It started off very simple with our G protein or our ligand gated, okay? And now we're just adding things on to show how we're going to get that cellular response in the end. Okay. So where's the response going to be? Could be in the cytoplasm, could be in the nucleus, okay? It could be anywhere, It doesn't have to be outside of the cell. It could be inside the cell. Most of the time it is going to be inside of our cell. So once we get our phosphorylation cascade, this one, we're going into the nucleus here. Okay. So whatever response it is, we're going into the nucleus. We are activating this transcription factor. And what this transcription factor is going to do in this example is going to convert our DNA into mRNA. So all of that, this could have been testosterone, this could be whatever hormone out here, we're bringing it in, we're going through using our kinases and all of that. And then the kinases down here, our response will be to activate our transcription factor. And then our goal for this one, I'm guessing in the end, would be to make a protein, since we are converting it from DNA to mRNA, because usually our next step from mRNA is going to be translating it into our amino acids. All right, we are good here. Last thing about this, guys, is these signaling mechanisms. These signaling mechanisms has been around for a very, very, very long time. We've noticed some of the most ancient prokaryotic cells have these same exact signaling mechanisms as our eukaryotes do these days. So this tells us once again, through an evolutionary sense, that, hey, yeah, we might have had all a common ancestor at one time if we go back far enough. All right, guys, that is the end of chapter 5. Keep working on your guided reading and have a good rest of your day.